Hi, my name is Katherine. I'm a postdoc in the Danforth Lab. I'm currently working on the interaction of native bees and their brood parasite. And I'm currently running experiments right now with the host in this little jar right here. You might be able to see the little bee with her pollen on her bright yellow legs. And what we're doing is seeing if she can choose between an arm that has her conspecific, so her another individual of her same species, and just empty air. And this is important because it tells us whether or not these bees might be using chemical cues from others like themselves in order to make large nests. So this research was based upon a paper that was written in the 1960s, and they were trying to figure out how it is that these parasites can enter the nests of their hosts and remain undetected. We've come so far, at least in the realms of chemical ecology and entomology, where those results probably are not correct, and we are just running our experiments to validate or disprove those that paper. But we're really lucky here at Cornell and just living in Ithaca, where we have a lot of these large bee cities, or large, what we call aggregations of bees that attract their brood parasites. Because of this, we've started documenting potentially new behaviors in these bees, and it just helps add to our knowledge of how to conserve them in the environment. So I got into entomology as a very young child. I took a class at the museum from my hometown during the summer, and it was all about bugs. And I just thought they were the coolest thing. I did my PhD work with brood parasitic bees, particularly this genus called Nomada, or the nomad bees. They're my favorite. They're the most diverse of all of the brood parasites. And that eventually led me here to Cornell to continue working on and expanding my brood parasitic bee research.